Welcome to Chapter 43, Animal Nervous Systems. By the end of this chapter, you should be able to describe the mechanisms in neurons that establish and maintain membrane potentials. You should be able to explain the steps in an action potential. You should be able to explain the events that occur at a chemical synapse when a neuron communicates with another tissue. And you should be able to describe the structure and function of the components of the vertebrate nervous system. All right, so the central nervous system includes the brain and the spinal cord. So the brain and the spinal cord. And the job of the central nervous system is to integrate information. Okay? So sensory neurons will collect information and send it on, and then interneurons, which are part of the central nervous system, pass the signals from one neuron to another neuron. So this is a neuron that's on the inside of your spinal cord. Some interneurons connect to motor neurons, okay, over here, and these are nerve cells that signal effector cells in glands or muscles to do something. So we'll get to those in a minute. Motor neurons and sensory neurons are bundled together in long strands called nerves. Okay, so the central nervous system is your brain and your spine, right? Spinal column. The peripheral nervous system, PNS, includes the components of the nervous system that are outside of the central nervous system. So you have sensory information that is transmitted by the peripheral nervous system to the central nervous system where it's processed. And then a response is transmitted back through the peripheral nervous system to the appropriate parts of the body by motor neurons. So for example, if you touch this spiky rose here, then that pain is detected by a sensory neuron, part of the peripheral nervous system. That stimulates the interneuron over here in the spinal cord. And then the interneuron stimulates motor neurons that affect the muscle, that cause you to withdraw your hand from the thing that hurt you. Okay, so a neuron has usually three parts. The cell body, or the soma, is the big fat part over here, and that includes the nucleus of the cell. There's a highly branched group of little projections called dendrites that come out from the cell body here. And then there's one or more relatively long projections called an axon, okay? So the dendrites, these little fluffy things, convert chemical signals to electrical signals. The cell body integrates the incoming electrical signal and then sends it along the axon to the next cell. So the neurons will form networks for information to flow from one place to another. I said that. All right, which of these choices is the correct direction of information flow in a neuron? Where does it go first?
The dendrite.
All right, then what?
Then it goes through the fat part of the cell.
What's that called? That's the cell body. Yeah, it goes through the cell body. And then where does it go?
That long skinny part.
All right, what's that called?
Oh, that's the axon.
All right, so it goes dendrite, then cell body, then axon, right?
Yeah.
All right, so we're going to talk a little bit about membrane potential. And so this is the difference in charge on either side of a plasma membrane. So if you have a lot of positive charge on one side of a membrane and negative charge on the other side of the membrane, this creates potential energy that can do work. And so that's called membrane potential. All right, so let's just say that you have a lot of sodium over here and a lot of potassium over here. Which way would the potassium go if it could?
What's this picture showing?
Well, that you have a lot more of the potassium over here and not so much over here.
Oh, so it would want to go up.
Yeah, it would want to go up. What about the sodium?
Oh, it would want to go down.
Yeah, it would want to go down. Okay? So that's potential energy that could be harnessed. And the way that it is harnessed is by using a sodium-potassium pump. And the way that your book abbreviates it, it's sodium, potassium, ATPase, right? When I read this phrase, it kind of makes my brain hurt, but you kind of get what it's saying. So this is a sodium and this is a potassium, and the ATPase is an enzyme, that it is basically making ATP, okay? So what we have going on here is we have a sodium-potassium pump, right? So if you have sodium that goes in here, and a molecule of ATP that goes in here, it can use the energy locked up in that ATP to transfer the phosphate from the ATP to this pump, okay? And so then that ATP it's basically going from being a charged battery to being an uncharged battery. It's transferred its 1-phosphate over here, and that energy is used to pump 3 molecules of sodium across the membrane. And when it does that, it allows 2 molecules of potassium to enter, and so then those can go in this way, and when the 2 molecules of potassium come back inside, that releases the molecule of phosphate, which allows this pump to work again. So this process basically puts 3 molecules of sodium over on this side of the membrane and brings in 2 molecules of potassium, okay? So this pump is creating a gradient where it has a lot of sodium over here and a lot of potassium over here. So you get a lot of potassium inside of the cell over here and a lot of sodium outside of the cell. So again, the pump establishes an electrical gradient. So each time it pumps, 3 sodium out and 2 potassium in. And so the inside the, well, probably the word cell is missing. Inside the cell is less positive, right? More negative than outside, just because you end up basically putting 3 sodium out for every 2 potassium that come in. And so the relative charge of outside of the cell versus inside of the cell is that outside is more positively charged and the inside is less positively charged, so it's kind of like the inside is more negative and the outside is more positive, okay? So now you have a difference in charge inside and outside the cell, so that's membrane potential. All right. So an action potential is a rapid temporary change in the membrane potential. So remember we talked about how when it's at rest, just show you again, when it's at rest, the outside of the cell is more positively charged than the inside of the cell. The inside of the cell is more negatively charged, okay? So we see here that at rest, the resting potential is negative, right? It's around, I don't know, minus 70 millivolts, okay? And then something happens to depolarize the neuron. So at that point, it becomes so more and more positive until it's actually positively charged, okay, so less and less negative, and it moves towards being positively charged, and then it repolarizes, so it rapidly goes back to being negatively charged, and it'll even undershoot where it started out, and it'll be even more negative than it started out, and then it'll return to its resting potential over here. Okay, so that bit where it overshoots, or I guess you could say undershoots because it becomes a little more negative, is called the hyperpolarization stage. It's important to note then that when you have this change in the membrane potential of a neuron, it is an all or none event. So you can't have part of an action potential. Also, for a particular neuron, all the action potentials along that neuron are going to be identical in magnitude, meaning how much the depolarization, how big the peak of depolarization is, and also duration, how long it takes it to get from resting to resting again. So these action potentials are propagated down the length of the axon. Do you remember what the axon is?
Yeah, that's the long, skinny part of the neuron.
Yeah. Okay. So let's go over it step by step, okay? So here's your plasma membrane around the outside of your neuron, okay? And here's your sodium-potassium gates here, all right? So right now they're closed, okay? And at the resting state, the outside of the cell is much more positively charged than the inside of the cell, okay? And so the inside of the neuron is about 70 millivolts less than the outside. So that's the difference between inside and outside. So if you look kind of here, we are at negative 70 millivolts as the resting state for this particular neuron. All right, so then the sodium channels will open, and then the sodium ions are allowed to diffuse into the cell, okay? So this movement of positive charge into the membrane makes the membrane potential more positive, meaning less negative. So this is what's going on over here, okay? But at this point, the potassium still can't go anywhere, and we only have a few of the channels open, so that's the early depolarization part of it. All right, next, the third part where you have this rapid peak, the thing that happens now is that sodium is rushing through open channels, and that causes a rapid depolarization of the membrane, so the membrane actually becomes positively charged at this point. So we have lots of these gates opening. And then at this point here, at the top, the thing that kind of changes it from this depolarization to repolarizing is that these voltage-gated sodium channels inactivate. So the sodium is now blocked, and then the voltage-gated potassium channels open. And so now all the potassium diffuses out of the cell, and then that causes the membrane potential to become negative again. Okay, so that's getting you down here. And so these gates will remain open for a long time, and they'll actually remain open long enough that you end up going below the minus 70 millivolts that you started out at. Okay. Oh, should I say anything else? And then you're back to normal again. Sorry. Now then afterwards, you hit back at minus 70 millivolts, and you're back to your resting potential. Okay. And this kind of depolarization and then repolarization is happening all along the length of the neuron, okay? So you can use, this is just a picture, these little carrots or electrodes, right? So it's just measuring, if you had a way of measuring what was happening here on the neuron and then here, what you would first see is that over here, as the information is being passed from the dendrite through the cell body to the axon, right? When it hits here, this part of the neuron depolarizes, okay, and then comes back, and then that sends a signal on further on down the axon, and when it gets to here, then this part depolarizes, and then over here, this part depolarizes. One thing you might note is that this isn't like a ripple effect where once it depolarizes here and then it causes left and right to depolarize. It only goes in one direction, okay? And this is because once the sodium channels have opened and closed, they have to stay closed for a short period of time before they reopen. So that's called the refractory state. Because this part of the neuron has already had the channels open and close, right? When this part of the neuron depolarizes, it doesn't send the wave backwards, it only sends it forwards to the sodium channels that haven't yet been opened. So we say that action potentials propagate in one direction because the upstream, right, the ones before, sodium channels in the direction of the cell body are in a refractory state. So that's pretty much what I just described. All right, so what happens when you get to the end of the axon, okay? So the end of the axon contains synaptic vesicles. So we talk about the fact that one nerve goes all the way up to the next nerve, right? But they don't actually touch each other. There's a little tiny skinny gap, like you can see it's very little, but one nerve cell doesn't touch another nerve cell. Instead, they communicate across this very thin gap called a synapse. So this is the end of one axon, and then over here is the dendrite of the next nerve, okay? And so the end of the axon contains synaptic vesicles. So what's a vesicle?
Yeah, that's like when you got a little bubble of something that gets carried around inside the cell.
Yeah, it's like a little bubble of something. And in this case, it's carrying neurotransmitters. So these are molecules that convey information from one place to another. So the vesicles, these little circles here, will fuse with the synaptic cleft and dump their contents, which are these neurotransmitters, into this gap. And then the neurotransmitters go across this little gap until they get to the next dendrite. And then they bind to receptors over here in the dendrite. So that's how the information or the electrical impulse that's over here in this axon is converted into a chemical message that gets received by the next neuron. So here's like a schematic showing it. Okay, so here's one axon, and then it's this over here is the dendrite. So here's the axon of the one nerve cell, and here's the dendrite of the other cell. All right, so here we have the sodium and potassium channels, right there. And so first the action potential arrives near the synaptic cleft, and so when that's happening, so you have the sodium and potassium channels opening and closing, so the action potential is working its way down the neuron, okay? When it works its way down to the end, you have voltage-gated calcium channels, right? So these are channels that can allow calcium in, basically, but only if they are stimulated by electricity. So when the action potential gets all the way down to the end, that stimulates the calcium channels to open, Okay, so now the calcium channels are open, and that causes the synaptic vesicles. So this is a vesicle filled with neurotransmitters. So when the calcium channels open, it causes the synaptic vesicles to fuse with the edge of the axon. Okay, when they fuse, they kind of fuse their membrane and then dump the contents, all of the neurotransmitters, into this cleft, this gap between the axon of the one cell and the dendrite of the next cell. All right, over here on the dendrite, there are receptor, little receptor molecules, and the neurotransmitter will bind to these receptor molecules. Okay, and when the little receptor molecules bind, this causes a change in the membrane potential of the dendrite of the other cell. And so that causes the information, the electrical information that was conveyed from the other axon to start to be propagated through the dendrite over here into the other nerve cell. After the information, the electrical information is passed down through the dendrites, these ion channels in the postsynaptic membrane close up, and then the neurotransmitters unbind. Okay, so then everything kind of resets and waits for the next action potential to arrive. All right, so imagine you are being chased by a bear.
Ah!
Okay, I guess you have a really good imagination.
Yes, I do.
All right, so your body is likely to experience many physiological changes in response to this stressor. Name as many physiological changes as you can think of.
I would probably be breathing really hard.
Yeah?
You would probably be sweating too, but not me. Birds don't sweat so much.
Yeah, Bird, what would you be doing?
I would be getting like a rush of adrenaline.
Yeah, you get a rush of adrenaline, right?
It would make me feel really nervous.
Yeah? What else? Anyone else?
I'd get a really dry mouth.
Yeah, you get a dry mouth because you're scared?
Yeah.
What else?
I bet he'd poop himself. Yeah, I might poop myself.
Yep, that's another thing. Okay, so those are all the kinds of physiological changes that you might feel if you were being chased by a bear or had something scary happen to you. So let's talk about what's going on with that a little bit. So we have a set of nerves called the parasympathetic nerves, and these are the kind of nerves that are operating when we're just chilling out, when we're not stressed out and a bear is not chasing us. So for example, another way of thinking of the parasympathetic nerves is, kind of our rest and digest mode. So normally our pupils are, constricted, we can salivate, we have a relatively slow heart rate, our airways are, kind of constricted a little bit, we are able to digest food, our liver is not secreting extra glucose or stimulating the gallbladder. We have kind of stimulated activity of the intestines, so our digestion is occurring. Our bladder is usually kind of contracted, and under these circumstances, if the mood arose, we could be reproducing or whatever. Okay. However, when a bear is chasing you, this stimulates a set of nerves called the sympathetic nerves. And so this is what we refer to as the fight or flight mode. And so when this happens, if something was terribly frightening to you, your pupils would dilate. That would potentially allow you to take in more light, your salivary system dries up, so your mouth gets dry, your heart rate goes up, your airways become dilated, which will allow you to have higher aerobic capacity. The effort that is being put into digestion, is stopped. So your digestion basically stops. You get a stimulation of the secretion of epinephrine and norepinephrine from your adrenal gland, which makes you feel really jangly and rushed and everything. Your bladder is relaxed. You might pee. This is, for whatever reason, also associated with promoting ejaculation and vaginal contraction as well. So this would be Sympathetic nerves, fight or flight, versus parasympathetic, rest and digest. All right, so hopefully in this chapter you learned how animals use electrical signaling along neurons for internal communication. You learned the principles of electrical signaling, you learned how action potential works, you figured out what happens between neurons, and then we just really briefly talked about the vertebrate nervous system. We didn't talk about the brain or learning or memory, so don't worry about that.